Right, we are ready to go. Are you ready, sir?
Yeah, sure, sir.
Just give me 1 second and start. Good evening, everybody. Welcome to this evening episode of cGRIP, which is Career Guidance for Residents in Pathology. And we have a very, very interesting topic today, which is specialization in cytogenetics, molecular genetics, opportunities, and prospects. I think this is the subject which is the way forward. And to talk on that is somebody who is an expert in cytogenetics, Dr. Mayur Parihar. He is an MD and fellow in cytogenetics. He's the head of Kolkata. He's post CMC Vellore. Before I start, request all of you keep your mic muted, camera off, and please join your request Dr. Parihar, sir, please share screen and start. Thank you so much.
Thank you, Dr. Nadeem, for giving me an opportunity here to share my experience. I'll start by asking the most frequently asked question in today's times. Can you all see the screen and hear me?
Yeah. We can all see your screen and hear you. Just press that hide so that
the block goes away. Yeah.
Yeah. Thank you so much. Please start.
Thank you. So it was interesting, when Dr. Nadeem called me and said, "Would you like to speak on this?" And I must say it was a little difficult topic because you have to basically discuss your whole experience in what you have been through when I took a call to do a fellowship in cytogenetics myself. So just like most of the residents, when I had just finished my MD, I was wondering after MD pathology, what should I do? And I really remember very well that one of our professors in AFMC told me that, Mayur, once you finish your MD path, just take your bag and go on a pilgrimage to 4 institutes in the country. Go to CMC Vellore, go to PGI Chandigarh, go to TMH Mumbai, and go to AIIMS Delhi. And just try to get into any one of these institutes in any branch or field or subspecialty of pathology you can get into, and you can then take your career ahead from there. But I was fortunate that I could make it into TMH TMH Mumbai, where I did my SRship in histopathology for some time. So that is where my journey started after MD Path. But at the same time, I would like you to share a story of a friend of mine who was also doing his MD from AFMC. And what happened was he went on to do a 3 years SRship in one of the premier institutes of the country. Now, the SRship pattern there included a complete rotation in different subspecialties of pathology. So he had, say, 6 months in transfusion medicine, 6 months in histopath, 6 months in hematopath, 6 months in cytology, and so on. And after 3 years, when he went on to take a job in Delhi, people asked him, "What are you? Are you a histopathologist? Or are you a hematopathologist? Are you a nephropathologist? What is your specialty? Are you a cytologist?" And then he realized that it was difficult for him to get a well paid job, because this is almost 10 years back when everyone was asking about what is your specialty. And I'm telling you this story to bring about and reiterate the fact that subspecialty training today has gained a lot of traction in pathology. So you could choose any branch of pathology as a subspecialty, including clinical pathology, which is itself is a big branch of pathology today. You could even go into management if you want, right? Managing the laboratories. You could take histopathology, cytopathology, and of course, today we have cytogenetics and molecular genetics. So finally, this friend of mine went and did a postdoctoral certificate or a fellowship course in nephropathologist, and today he is a very famous nephropathologist. So although I'm sure working for those 3-4 years helped him build a good platform to launch himself, end of the day he really had to train himself in a subspecialty and in order to find a good job. So today I'll be talking about the subspecialty in cytogenetics and molecular genetics. What are the opportunities? What are the prospects? And so on. So to start with a very basic concept, what does a cytogenetic department do? So it does study structural and chromosomal abnormalities. It does what is called as karyotyping and FISH. And a molecular diagnostic lab would actually go deeper into the chromosome. They look at the DNA, the RNA, they do PCR, and a lot of other molecular techniques. But I must say that as time is going by, the line between the two is disappearing, and the fields today are merged as genomics. And as we go ahead, within these fields also, there are subspecialties in which people are expert in specific aspects of either molecular diagnostics or cytogenetics. So this is what a cytogenetic lab does, or a cytogeneticist does. He looks at the chromosomes and he identifies the chromosomes and looks at the different banding patterns. So it is just like your morphology, wherein you look at difference in different banding patterns and you're able to actually make a diagnosis of identify an abnormality which is consistent with the diagnosis. Or also identifying these abnormalities today help in risk stratifying various tumors in modern treatment protocols, right? So karyotyping as a tool is gradually going away in constitutional abnormalities, wherein you have SNP array. So our cytogenetic lab today does SNP arrays as well, and they try to identify abnormalities in constitutional in children or people with constitutional abnormalities. The other thing a cytogenetic lab does is FISH, or fluorescent in situ hybridization, wherein it's a very simple technique. You have sequences of DNA that are tagged to fluorescent probes, and these go and hybridize to your genes of interest. So you can actually look at the genes through your eyes under the microscope and make a call about, are there translocations, are there deletions, are there fusions, or are there amplifications, and so on. But believe me, doing all this, you have to use your basic pathology knowledge. And what test to be, how do you interpret your karyotypes, or how do you read your FISH? A lot depends on your basic morphology. For example, if you're doing a solid tumor fish, you need to know your histopathology well. You need to look at the slides. You need to identify the tumor areas. Otherwise, what you will do is you would probably only count cells like the supporting, instead of the tumor cells, cells like lymphocytes or the stromal cells, and you would actually miss the abnormality that is seen in the tumor. So your basic pathology or your morphology always stays, and this is on and on this you build upon these novel techniques and integrate the two. And believe me, it is great fun to go from morphology to immunohistochemistry or immunophenotyping into the genomics, and then try to unravel the different abnormalities, the tumors, the diagnosis, and so on. What does a molecular genetic lab do? It takes the DNA and RNA, and then it uses a host of techniques. I won't be going into technical details because today's talk is about what are the prospects and career opportunities. So you can do sequencing, which could be a first generation like Sanger's. You could do next generation sequencing. You could do PCRs or gel pictures. So molecular genetics in a molecular genetic lab, based on the question that is being asked in terms of the diagnosis, in terms of the clinical context, in terms of the pathological context, you then take a call and choose a specific molecular technique that would be the most efficient to identify the abnormality you are looking for. At times, yes, you do go on an exploratory mode, and therein you use more generic whole genome or next generation or sequencing techniques wherein you try to look for the unknown abnormalities. So many a times in this field, you do don the cap of a detective, and many times you see subtle abnormalities like you see on this karyotype here. It appears that there is something wrong with 11 and 19, but you're not sure, and you're thinking there's something wrong, kuch to gadbad hai. Yeah, and then you put additional fish probes, or you use molecular techniques. You correlate them with flow cytometry or immunophenotyping, just like the quintessential detective from this part of the country, Byomkesh Bakshi, and actually you solve the puzzle to make a diagnosis and identify the abnormality. So unlike Byomkesh Bakshi, usually in my department we are no Kesh Bakshis. That is Arun, who's a scientific officer. And believe me, it is great fun. You look at the slide, you make your diagnosis, you look at IHCs, or you look at the flow cytometry of the hematological malignancies, go back at the karyotype or the fish, looking at the molecular mutations, and then take a call, integrate all the possible knowledge, and make an informative call on the diagnosis. But you must understand that suppose you take this as a career option, you have to be first a technologist. You have to spend significant amount of time on the bench. So be it cytogenetics or molecular genetics, you learn from the technologist. So you have to be on the bench. You should be able to make your own reagents. You should be able to run everything. So because as it is in India, what happens is when you get trained, there are two aspects of getting trained in any field. One is to acquire your training from a good place, and second is to find a place that lets you apply your training. You may train in the best institute in the world, but post your training, if you do not get an institute with an infrastructure where you can apply your training, soon you would actually forget what you have learned. So many times, what would happen, at least now, is you may have to establish a complete lab from the scratch. So for that, you have to know everything. So when we started the lab here, I was able to make all the 1 molar solutions or 1N solutions. I could make all the reagents, and even today I can run the lab myself. So you have to be a technologist, get back on the bench. Yes, molecular and cytogenetics looks very glamorous, but believe me, it is very tedious, it is laborious, it can be repetitive, cumbersome, but at the end of the day, when you get the results, it is really fun working on it. So if you're thinking of coming into this field, do not think that you would be sitting on a chair and you would be just looking at some chromosomes, or you would be just interpreting some plots or analyzing genomic data on your computers. No, not at all. You have to be on the bench and learn the techniques so that you can troubleshoot as and when it is required. Now, you need to understand that if you want to pursue a career, a full-time career in this field, you need to get completely trained. There are no shortcuts. You cannot go for a 2 weeks training and become an expert, right? You could. There are fellowship programs, but if many of who are now feel it's difficult to go back to do a fellowship for 2 years, what you could do is you could spend at least 3 to 6 months and learn certain techniques, and then come back, try to apply in your place of work, and gradually build upon it. But the devil lies in the details, but that is where you will also find salvation, right? So there is no shortcut. You have to really spend a lot of time in order to master these fields. So there are fellowship programs in the country. CMC Vellore was one of the first to launch. You have fellowship programs in molecular genetics, and you have fellowship program in cytogenetics. These are 2 year programs, and yes, you do get paid in this fellowships. TMC Kolkata, again, we have a 2 years program in molecular genetics and cytogenetics. I can proudly say that in TMC Kolkata there is a very free flow of information. So even if you are doing a cytogenetic fellowship, you could always spend your time, spare time, and go to the molecular labs and other ways. And we do post our fellows in both the departments, so that a fellow is trained in both, but then they make a choice, that I want to be an expert in molecular genetics or an expert in cytogenetics, but as well, yes, be able to both run and interpret the other techniques. Here I would like to say that cytogenetics has a much long learning curve, because it is a lot of morphology and it will take you a lot of time. Molecular genetics, on the other hand, does have a learning curve, but but you can pick it up quickly, but sky's the limit there. Yeah, right. With so much of genomics data being available today because of high throughput sequencing techniques, you need to learn how to analyze this data. And even today, I am learning. I am learning computer programs. I am taking classes as how to use informatics, how to do some coding and try to analyze this data. Because even if you have somebody who's an informatician and helping you with that, he will take you for a walk in the garden if you do not know the concepts. So once you train in these concepts, you can tell your bioinformatic guy who is coding for you to actually tell what to do. So believe me, it's in these fields you have to, like any field in medicine, you have to keep upgrading yourself as time goes by, or you would actually soon be left behind. TMH Mumbai has a fantastic molecular genetics program again. So CMC Vellore usually the fellowship starts in August. So you should look in these websites by March, April, and you would have the fellowship. And similarly in TMC Kolkata also. And TMH Mumbai comes around the time of October, but you need to keep a track on your website. So usually most of these all we have entrance examinations which are MCQ based, following which you have an interview and you would go for that. Uh, I always feel it is always good if you have. It's always good if you have already worked in a field of pathology before coming into molecular genetics and cytogenetics. Because sometimes what happens as a fresh MD path, when you really jump into these fields, some of them, you could start feeling insecure. Am I forgetting my hematopathology, morphology? Am I forgetting my histopathology, morphology? So I think the best candidates are those who finished their MD path, worked for 1 year into one of these fields, and then made up their mind, yes, this is what I want to do, and then you take the plunge. A half-hearted attempt on these will actually be really difficult and will leave you more confused rather than show you a direction in your careers. Now, once you've done this fellowship program, see, be it cytogenetics or molecular genetics, where do you apply the knowledge you have acquired? Now, each of these fields, you could use them in constitutional disorders. And when I say constitutional, it's like children with mental retardation, children with birth defects, rare gene disorders, single gene disorders. There are a lot of benign hematological diseases where This can be involved. There are storage disorders, and today in our country, in each of these fields, we have experts. We have experts in only thalassemia genetics, right? You have experts in only sickle cell anemia genetics. You have expert on lysosomal storage disorders genomics. So as I said, there are a lot of fields you can actually then take on and become an expert. Expert there. It's all up for grabs, and I feel so far the pathologists have stayed away from cytogenetics, molecular genetics, and that is the reason we've not been able to get into these fields, and we believe that the basic biology people will do a much better job. But I think with our training and knowledge, once you get into it full time, you can really do as well, or rather even better than the others, right? And of course there is cancer genetics. So like for example me, I specialize in cancer genetics, right? I have an idea of all the other techniques and disorders, but I'm not an expert in that. So I'm an expert in cancer genomics, and that is how you can apply these knowledges where you are. Now, a big question that comes into the mind of most of the residents or people who get into this: does doing this fellowship means that you stop practicing other aspects of pathology? So you may be a histopathologist from training, a hematopathologist or biochemical pathologist. No, it doesn't mean you stop practicing other aspects of pathology. You, in fact, build upon it. You integrate your knowledge of histopath or hempath. But one must remember that you cannot do everything unless you are Leonardo da Vinci, right? You will have to take a call. For example, if I give my own example, I do hematopathology, but I just look at bone marrow aspirates and biopsies. I don't do high-end flow cytometry because a lot of my time in the day goes into molecular genetics, to cytogenetics and molecular genetics. So if you ask me, okay, you do hempath, are you a flow cytometry expert? No, I am not. I cannot be everything because there is just that limited time, and each of this field is growing at such a fast pace. So you could actually choose your area and then build, get this training and integrate this knowledge with your existing knowledge, and then you would be able to apply that in a more objective manner. So I've known people who are histopathologists, but then they say, okay, I would do soft tissue tumor genetics only, and I would be an expertise in that. That doesn't mean they don't know about others, but that's how they go on to become experts. But I think today in India, we do need people with a much more broad-based, much more broad-based, wherein they have knowledge to apply in all of these. But as I said again, doing this does not mean you completely stop your histo or hempath, but you will have to take a conscious decision as to what you want to pursue, because you cannot pursue complete everything, as I said before, unless you're Leonardo da Vinci. Now, what are the job opportunities? There are a lot of job opportunities in academic institutes. Now, a lot of medical colleges in the country are getting cytogenetic and molecular genetic infrastructure being, funded by the government. In fact, all AIMS have today a cytogenetic and a molecular genetic setup, but they do not have people who are trained to apply this knowledge. And we've been training a lot of people as observers from these institutes to actually come and train with us and go back and apply the knowledge there. So there is a big demand today for people trained in cytogenetics and molecular genetics, both in academic institutes and AIMS. It's sad, but yes, it's a fact also that the corporate labs today have much bigger and better infrastructure than academic institutes. So there are a lot of job opportunities in corporate labs wherein you will get the best of the infrastructure. And if you are someone who would like to go the corporate way, you can actually, there is a huge demand. Every week I get a call from different corporate labs. Do you have somebody trained? Do you have somebody trained? All across the country, in fact outside country as well, be it Malaysia, Gulf, and so on. Can you send somebody? Do you have somebody whom you can train? And we have actually trained people in our lab from Malaysia, Egypt, Saudi Arabia. So there are a lot of opportunities there too, wherein you can actually train and go into this. The other opportunity is that once you master these techniques, you could actually help you do a lot of research, right? So you could write research funds, research grants, and get funding. So if you are a histopathologist, you want interest in soft tissue sarcomas, you could actually write research grants on soft tissue sarcomas, integrating that with genomics, developing new techniques that can be used in India, and and you could actually build upon strategies that can be built in resource limited settings with all of your knowledge and follow research. And there are a lot of funds with research agencies in India, and you come up with a good project, you and you are asking a good question, you would get your research funding. So I always believe in life, you would always get the right answers if you ask the right questions. So that's how it is in research. Second, there are opportunities abroad itself. As I said, Gulf, you could try to go into Europe. Each of these countries have their own set of licensing exams, which could be tempting. But you could straight away get a job as a research postdoc in any of research labs outside India. I know some of my students have done that, and they are now doing a postdoc in research, and they've taken that as a career. So it gives you a different dimension, to take your career ahead if you go into these fields. Now there is an American Board of Medical Genetics. So in some of the medical institutes in America, without an USMLE, with your credentials, you could get a ACMG waiver. So you could get an ACMG certification. Like I got one for myself. With that, I could apply for fellowships in different institutes in United States of America. So that can be done as well. Just giving you an idea of the journey. This is my fellow Umang Patel, and so this is a board we have outside our lab, and we just started with initial few publications, and this is just last year when the new fellow, and you can see how the board has got filled up. So there is a lot of exciting things in this area that you can actually publish a lot of papers, because you keep finding new things, and there's a lot of new data that keeps coming up. So that's an opportunity. So just giving you an idea how in last 10 years we've been able to publish in some of the top journals of the world, with great data. And this has also given us opportunities to build strategies for India, because you cannot follow the strategies in the West with our limited resources. So you come up with your own strategy wherein it is cost efficient, but you're giving all the information. So this is where you can actually contribute back to the society and to your country. So we've built a lot of these strategies which are working pretty well for us. I just thought I will summarize what my students have done. So one of my students, he's in US doing a postdoc. The others are here. Dr. Anurag, he heads the Empath lab. Umang, he was from Gujarat, so he set up his own cytogenetic and molecular lab, and he's been making a lot of money, let me tell you. Ashish is with us now as a clinical associate at TMC Calcutta. Dr. Manish is an assistant professor at SGPGI. So SGPGI really wanted somebody to set up their cytogenetic unit, and he just finished his fellowship at the right time and was able to get in there. Manish had job offers from Israel and UK, but he was in love with Lucknow, so he just wanted to be in Lucknow. He's moved in there. Tanvi is now in charge of the labs in Lalpath Lab, Kolkata, and Lieutenant Colonel Barun now is developing this facility in AFMC Pune. This field would also give you an opportunities of a lot of international collaborators. So, as I said, sky's the limit. We were able to collaborate with some of the biggest names in the world. So this is Professor Anthony and Oscar Haas, whose papers we have read when I was a student. And then it came in to contact through conferences, and then they visited our lab, and we worked with them. And we've been able to take this international collaborations much ahead. This is when I visited Vienna, where I not only learned genomics, but I also learned how to appreciate good beer. So there are a lot of opportunities, and as this is such a niche field, now we are a part of an international array group, and you can really actually go out and the data we are producing in India, because we have such big numbers, is fantastic, and you will get opportunities to present this data all across the world and impress everybody. So I would like to end by saying this is one of my favorite quotes from one of my favorite movies, Kung Fu Panda. Those who have not watched it, please watch it. Where Master Shifu says, "If you only do what you can do, you will never be more than who you are." So with that, I would like to thank all of you for your patience here. And if there are any questions, I am happy to take.
Uh, Thank you, Dr. Mayur, for this very interesting and very eye-opening presentation. If you could just stop presenting so that we can see you.
Okay, I've stopped sharing.
Yeah, that's
Yeah.
fine. That's great. Yeah, perfect. There is one, I won't say question, but both are comments rather on the chat box. You can see Mariam Raza, she writes, please can you start series of lectures to teach cytogenetics?
Oh
And
yeah, sure. I would be happy, and think we were just discussing before this that Dr. Nadeem plans to have this series, and we would be more than happy to take it ahead for sure.
Rashmi Patel says, thank you, sir, for such a nice lecture as usual.
Thank you.
Yeah, a lot of questions and comments will keep pouring in. As I told you earlier, also that all of it comes from the Telegram group. This is watched across overnight because we understood about 2 years back when we started this program of teaching online that everybody is not available at a given point of time.
Yes.
So we have to have, and you are not available every time. So
Yes.
the expert is available only on a given point of time, and the people who are going to benefit from the experts are also busy in various aspects, but they still want that to be benefited. So we decided to have everything open. So we have this YouTube channel whereupon we have this recorded versions. We have Google Drive where we upload all the PDF of lectures. This is not a lecture, but a general teaching lectures we upload. So maybe by tomorrow morning, more than 600, 700 people must have viewed. And maybe
Oh wow.
even 10% of them change their mind to think about cytogenetics and molecular genetics as a career. That's a great achievement, I would say. As I
Yes.
told you in the beginning before we were on live, that majority of the people do not know what is in store. So it's experts like you know, who really have gone through it from zero, can really guide them. And I'm so
Yes.
thankful that you consented. You are so busy. I know you are very, very busy. That you consented to take up this lecture,
Sir.
and maybe later I will talk to you about how we can, you know, think
Sure,
about what
sir.
Mariam Raza is saying. Series of lectures on cytogenetics. Maybe we will have one lecture a month or something like that, and maybe try to pro.
Sure, sir. Thank you. Yeah,
add-
the pleasure is all mine. I would be happy to contribute.
Um, I don't think so. I have any questions left because the way you have conducted this lecture and the way you have drafted it, I think you have closed on to everything. Everything, I mean, starting from exactly what you feel when you do an MD and how you go about, what are the aspects, prospects, which are the centers which offer, and how, when do they have their exams, what are the type of exams, and then what is expected out of you. And one thing which I really liked about your talk was the acquire and apply. That is the gist of everything. I mean, you have to acquire the knowledge and you have to have the opportunity to apply the knowledge.
Yes, sir.
And you have to be very, very lucky to have both. And you have been, and as you said on your pilgrimage tour, I mean, I think you only missed out PGI Chandigarh.
Yes,
The rest
sir.
of the four pilgrim spots you have already visited.
Yes, sir. No, so we
You're
have trained people from PGI to develop cytogenetics there. So,
great.
I have been able to pay my pilgrimage in a
Yeah,
different way.
homage to that place as well.
Yes,
True, true, true.
yes,
Yeah.
sir. Yes.
It was a wonderful, listening to you, Dr. Mayur. You're
Thank,
not only a excellent cytogenetic person, but you're a wonderful teacher. It's so nice to interact with you.
thank you so much,
Okay,
sir. Thank you for your
right,
compliments.
right. So I think we are done with the day, and thank you so much. I will get back to you. Maybe you can draft out a teaching protocol, and then we can find out a way out how you or maybe your associates can all be put in together, and we can do some justice on the teaching process.
Sure,
And
sir.
that would be a huge contribution to the society, to the fraternity, and I think you also understand the void which is there,
Yes,
right?
yes, sir, sure. So, of course, sure, sir, I would be very happy to work with you on this and take it forward.
Right. Thank you so much, Dr. Mayur. Thank
Thank
you.
you.
God bless you. Thank you so much. Thank you. Good night. Bye bye. Take care.
Good night. Thank you. Good
Good.
night.
Thank you.